નમસ્કાર, હું આદર્શ છું. વેલકમ ટુ આદર્શ એજ્યુકેશન સોસાયટી. Standard 9th, Subject Science, Chapter 5, The Fundamental Unit of Life. Dear students, in the last video we have discussed the osmosis and their activities. Now we will see that unicellular freshwater organisms. Unicellular means single cell. They are living in the freshwater like river, pond, lakes, and these organisms and most of the plant cells tend to gain water. Gain means what you are saying, getting water through the osmosis. Okay, absorption of the water by the plant's roots is also an example of osmosis. See, these two are examples. Thus, diffusion is an important in the exchange of gases and water in the life of the cell. So, diffusion is an important process for exchanging the gases and water in the life of a cell. In addition to this, the cell has to obtain nutrition from its environment. So, cell has to get the nutrition from its environment or from its surrounding. Different molecules move and in and out of the cell. See, it should, what you are saying, it should transfer from in and out through the type of transport, and for this transport energy is required. See, requiring use of energy. So that is in the form of ATP. ATP means adenosine, ADENOSINE, adenosine triphosphate, TRIPHOSPHAT, triphosphate. So energy getting in the form of ATP. So ATP is supplying the energy. The plasma membrane is flexible. Flexible means that move here and there and without breaking, and is made up of organic molecule that is called as lipids. Okay, lipids here. Lipid means fats and proteins, amino acids. See, plasma membrane is flexible and made up of lipids and proteins. However, we can observe that the structure of the plasma membrane only through an electron microscope. So we can observe the plasma membrane only through an electron microscope. The flexibility of the cell membrane also enables the cell engulf in the food. Engulf means what you are saying, covering the food and the material from its external environment. So you know that from the external environment, cell has to get the food. Such a process is called as endocytosis. See, from cell gets the food from the environment in the solid form. Such a process is called as endocytosis. And the cell has to get the liquid from the surrounding, that is called as phagocytosis. Okay, that process we are calling cell drinking also. Amoeba acquires its food through the endocytosis. Now we will discuss cell wall. Plant cells, in addition to the plasma membrane, have another rigid outer covering called the cell wall. See, after the plasma membrane, we will see the another layer that is called as a cell wall. The cell wall lies outside the plasma membrane. Okay, the plant cell wall is mainly composed of cellulose. Okay, what is cellulose? Cellulose is a complex substance and provides structural strength to the plants, and it provides strength to the plants. When living plant cell loses water through the osmosis, there is a shrinkage or contraction of the contents of the cell away from the cell wall. This process or phenomenon is known as plasmolysis. See, dear students, you know that whenever in the osmosis process, when water loses, the cell, if the cell loses water, so what will happen? Cell will get shrinkage or contraction or become smaller. Okay, then what will happen? The cell comes to the middle, the materials comes to the middle. For example, I will show you the diagram. If you see, this is the cell, okay, this is cell wall, and here this one. And when the cell loses water, so it shrinks, the cell shrinks, and this one materials will comes in the center, it comes away from the cell wall, away from the cell wall. Okay, this process is called as, what you are saying, plasmolysis. So this is also very important question. What is plasmolysis? We can observe this phenomenon by performing the following activity. Mount the peel of reho leaf in the water. So you have to take a leaf and you mount it on the slide. Examine under the microscope, powerful microscope. Note the small green granules you have to note this, that is contain chloroplasts, and these contain green substance called as chlorophyll. Put a strong solution of sugar or salt on the mounted leaf on the side. So what you have to add? You have to add sugar solution or salt solution. Wait for a minute and observe under the microscope. What do you see? So what you will observe? Cell loses water and becomes shrinkage, becomes smaller. Now place some reho leaves in boiling water for few minutes. This kills the cells. So what will happen if you will put the boiling water, okay, for few minutes? So then that kills the cells. Then mount the leaf on the slide and observe under the microscope. Put a strong solution of sugar and salt. Wait for minute, observe it again. What do you find? Did the plasmolysis takes place? And here what you are noticing, you are asking whether cell loses water or you will observe the plasmolysis. Okay, so you will not see the plasmolysis. Okay, what do you infer from this activity? It appears that as only plasmolysis takes place only in the living cells and the cell, not dead cells. See, you will not observe in the dead cells. So only the living cells are able to do the absorb water by the osmosis. From this activity we will come to know that what plasmolysis takes place only in the living cells. Cell walls permit the cells of the plants, fungi and bacteria to withstand very dilute hypotonic external media without bursting. See, if you, in the earlier you found that when you put in the cell in the hypotonic medium, what will happen? Water enters to the cell and it will become swelling and bursting. But here you see in the plant and fungi and bacteria cell, when you will put the cell in the water of hypotonic solution, dilute hypotonic solution, it will withstand with a longer time. It will not burst. In such media, the cells tend to take up water by osmosis. The cell swells and building up the pressure against the cell wall. See what will happen? Without shrinking, it built up the pressure to the cell wall. The wall exerts an equal pressure against the swollen cell. Because of their walls, such cells can withstand much greater changes in the surrounding medium than the animal cells. See, students, try to understand this. So you know that this is the cell wall. This is the cell wall. And here, this plasma membrane, and in this, when the water will enter from here, the cell will become swells and applying the pressure, equal pressure to the cell wall side also. So that is why this plant cell and fungi and bacteria, they can withstand without shrinking longer time. But animal cell, if you put in the for hypotonic medium, animal cell will die. Because why? Because The animal cell don't have the cell wall, but plant cell and fungi and bacteria, they have the cell wall. Okay, students. Now we'll move on nucleus. Remember the temporary amount of onion peel we prepared. So last time you have seen that which one onion peel. We had put the iodine solution on the field. Y is given. Y would see if the tried observing the field without putting the iodine solution. And try it and see what the differences is. Further, when we put the iodine solution on the field, did each cell get evenly colored? See, here students, what we would have done. So when you are observing the onion field under the microscope, we will put the iodine solution. And you have to observe both this. So onion cell without iodine solution also you have to look in the microscope. So you know that, dear students, when you will not put the iodine solution in the onion field or in the cell, so you know that cell is a transparent. So you will not get, you can't see clearly in the microscope. So that is why you have to put the iodine solution so that the cell gets colored, iodine colored. So you can study easily the structures. Okay. According to their chemical combination. So here given when you look, okay, when you have applied the iodine solution or when you have put the iodine solution, color what? You will see the same color, evenly colored is given. No. According to the chemical composition, different regions of the cell gets colored differently. See, when you put the iodine solution, you know that cells, lot of cells will be there, that will get according to the chemical composition that shows the color. Some regions appear darker than the other. Some are in the natural color and some regions appear darker. Apart from the iodine solution, we could then use we have used the iodine solution. Apart from this iodine solution, you can use safranin, SiFRANI, safranin solution also you can use, or methylene blue, METHYLENE, methylene blue also you can use to stain the cells, to color the cells. We have observed the cells from the onion. Now we will see the animal cell. Let us take a glass side with a drop of water on it. And what you have to do, you have to take using the ice cream spoon, gently scrape inside the surface of the cheek. Inside you have to gently move, slowly you have to move it. And you will get any material get stuck on the spoon. So you will see some materials will get stuck on the spoon. With the help of the needle, we can transfer this material and spread it evenly on the glass slide and kept ready for this. So observe. And to color the material, you can put a drop of, you know that, or any three, iodine or methylene blue or safranin. And here we have taken methylene blue solution on it. Now the materials is ready for the observation under the microscope. Do not forget to put a cover slip also you have to keep, because otherwise air bubbles will be entering. So what do you observe? What is the shape of the cells we see? And we will see that some dark colored spot in the cell or in the center of the cell. So that is a nucleus, and draw it on the observation sheet. Now, was there darkly colored, see here given darkly spherical or oval or dot-like structure near the center of the each cell. This structure is called as nucleus, where there are similar structures in the onion fields. So all the cells contains dark colored spot, that is called as a nucleus. The nucleus has a double layered covering called nuclear membrane. See, the nucleus has a double layered covering, that is called as a nuclear membrane. The nuclear membrane has pores which allow the transfer of materials from inside the nucleus to its outside, that is the cytoplasm. See what will happen, see the students here, see this one, and this is nucleus, and this from this areas, so this is covered by the cytoplasm, and this is with the pores. Here what will be there? surrounded pores will be there. So through this water has to pass in and out. So this is a, what you are saying, double layer, what, the transport of materials from inside and outside the cell. So that is to the cytoplasm. The nucleus contains chromosomes which are visible in the rod-shaped structures. So nucleus contains chromosomes which are what shaped structures? Rod-shaped structures. Only when the cell is about to divide. Only when the cell has to divide, then it becomes a rod-shaped structures. The chromosome contains information from the inheritance features from the parents to the next generation in the form of DNA. This chromosome contains information from the inheritance. From the inheritance means from the inherited from the parents to the next generation, hidden. This is stored in the DNA. So DNA is called as deoxyribonucleic acid. Deoxy ribo nucleic acid. See, chromosomes are composed of DNA and protein. Okay, what is the chromosome contains? DNA and protein. These DNA molecules contain the information necessary for constructing and organizing the cells. These DNA molecules contain information constructing the cell and organizing the cell. This information contains the DNA. And functional segments of the DNA are called genes. See, these functional segments of this one of DNA are called as genes. This also DNA contains. So information will be there in the genes. Chromatin material is visible as an entangled mass of thread-like structures. Entangled means, you know that entangled, like folded structures. Okay, chromatin material is visible in the entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes. So whenever cell has to divide, these entangled structures, or entangled means folded structure, that changes into, what you are saying, changes into chromosome structures. The nucleus plays a central role in the cellular reproduction. So nucleus plays a very important role in the cellular reproduction, the process by which a single cell divides and forms two new cells. It also plays a crucial part along with the environment in determining the way the cell will develop. So it gets from the environment chemical compositions and what forms will exhibit the maturity by directing the chemical activities and for the cell development. In some organisms like bacteria, the nuclear region of the cell may be poorly defined. Not poorly defined, you will not notice the nuclear region due to the absence of the nuclear membrane. So such an undefined nuclear region containing only the nucleic acid, this is called as a nucleide, Nucleoid. So nuclear membrane is absent in that cell. Such organisms whose cells lack of nuclear membrane, that is called as a prokaryotes. So we are calling prokaryotes. Pro means primitive, primary, or karyote means nucleus. So primitive nucleus. Organisms with cells having a nuclear membrane are called as eukaryotes. See, when nuclear membrane is there, that is we are calling eukaryotes. For example, which one? Prokaryotes example, bacteria, and you can say eukaryotes example, even human beings, you can say that. The prokaryotes is also lack of most of the other cytoplasmic organelles. So, other cytoplasmic organelles are also absent in the prokaryote cell, present in the, and eukaryote cell you will get all these structures. Many of the functions of the such organelles are also performed poorly organized parts of the cytoplasm. See, some of the structures, organelles are also poorly organized in the prokaryotic cell. The chlorophyll is photosynthetic prokaryotic bacteria is associated with the membranous vesicles. See, you know that the photosynthesis has to take place. So this in the prokaryotic cell, what will happen? For example, bacteria is given associated with membranous vesicles, but in the case of eukaryotic cells, this is filled with the plastids, okay, in the eukaryotic cell. So this is about nucleus. Now we'll see about the next, okay, now we'll discuss. See, these are the bacteria cell, prokaryotic cell. So you that say this is a nucleate. Nucleate, you know that nuclear membrane is absent, and when you will see the microscope, you will get these three important structures: ribosomes, plasma membrane, and cell wall. So many of the other structures are absent in the prokaryotic cell. So this is an example of bacteria. Okay, dear students, thank you. Thank you for joining us